Okay, so today we're going to look at the notes for DNA. So we're going to start off with the structure of DNA. As a nucleic acid, DNA is made up of several nucleotides put together. Each nucleotide has a sugar, a phosphate, and a nitrogen base. So if you look at the picture here, this is our nucleotide. These are the monomers for DNA, and they have a sugar, a phosphate, and the nitrogen base right here. And we know the nitrogen base could be one of four, which we're going to talk about in a little bit. Okay, so here we're going to talk about the shape of DNA. DNA is shaped in a double helix or twisted ladder manner. And if you look at this picture on the left, it shows you what a double helix looks like. Double because we have two sides. Helix because it's like if you grabbed a ladder and just twisted it, and that shape that it makes is called a helix. The sides of the ladder are made up of sugar phosphate backbone, while the steps of the ladder are comprised of the nitrogen or nitrogenous base pairs. This picture on the right shows you a little bit better what that's talking about. So here, this is one nucleotide, and then it's connected to another nucleotide, and then it's connected to another nucleotide. The outside would be considered the backbone, and that goes for the other side as well. The backbone, if you look closely, is made up of the sugar and the phosphate. These would be like the steps of the ladder. These are our bases. So the bases make up the steps of the ladder. The bases are matched in an anti-parallel way and are bonded by hydrogen bonds, which are very weak. So here you can see how they're not they're not really connected to each other. They're actually connected by these little dots. These are representing hydrogen bonds, and they're very easy to break. The nitrogenous bases and Chargaff's ratio. Here we talk about the four bases, which are adenine, thymine, guanine, and cytosine. Adenine is always going to pair up with thymine, and cytosine is always going to pair up with guanine. The way that I told my students to remember that was A goes with T, so you can remember the phrase apple on the tree, and then C goes with G, and so you can remember the phrase car in the garage. Now that makes sense, because if you say apple on the car or apple in the garage, it doesn't make sense. It only makes sense when you say apple on the tree. Same thing with car on the tree or car on the apple, or it doesn't make sense. It only makes sense if we say car in the garage. So that hopefully will help everyone remember that A goes with T when we're talking about DNA, and G goes with C. Because of this, Chargaff's ratio states that the percentage of adenine and thymine always match, and the percentages of cytosine and guanine always match, which makes sense, because if you have an A, you're always going to have it paired up with a T. So if you have 100 adenines in a strand of DNA, you're going to have 100 thymines. Likewise, if you have, however many guanines you have, you're going to have cytosines. Now, DNA replication. DNA must replicate before a cell goes through mitosis in the S phase, so that each cell can have identical genetic information. If we're making two identical cells, we want to make sure that they both have the same DNA, so we have to replicate it. We have to make a copy. And so DNA is going to be replicated in 4 steps. And this, I know this shows 3 steps, but step 1 and step 2 can be kind of joined together. So first, the double helix needs to be untwisted. And 2, we need to break it apart. We're going to need to break up the hydrogen bonds that hold the 2 sides together. And we're going to do that using helicase. So if you look at the picture with the number 1, you see this helicase, and we know it's an enzyme because it ends in ase, breaking up the bonds that were holding them together, that was holding the double strand together. 3 says, add nucleotides to each old strand using DNA polymerase. So on number 2, you can see that the old strand that was split apart by the helicase Now each one is having new nucleotides added to it. So this one has nucleotides being added to it here, and this one has them over here, and the enzyme that's doing that is DNA polymerase. And then finally, the nucleotides are going to get glued together using DNA ligase, because DNA polymerase only puts them on the strand, but we need to connect them. So we're going to connect them together using ligase. So that's another enzyme that's going to be responsible for putting the nucleotides together to form the two new strands of DNA. And this picture here shows us kind of what I just explained. And the reason why this picture is here is because you need to know that DNA replication is called semi-conservative because it results in two identical copies, each with one old strand and one new strand. So when we split up the DNA, this is part of the original strand, the old strand, and then over here this one is part of the old strand as well. Now the new one is the one that's being added. So this is going to be a new DNA, and this one is going to be a new DNA. So these are the two copies, but each one has a new half and an old half. That prevents errors from happening. If we try to copy this DNA strand just by adding new nucleotides to both sides, some type of mutations could occur. This will help prevent mutations. Okay, so those were the notes for DNA. Hopefully you're studying your top 10s ahead of time. The notes are already on OneNote. Make sure that you're ready for Friday. #Team Finesco.